start on chapter 10. Okay, chapter 10, which we're gonna study a special type or special class of molecules or biomolecules called enzymes. Okay, they are not a separate type of biomolecules. All enzymes are, in fact, proteins, which we have just completed in chapter 9. But enzymes are a group of proteins that catalyze all the chemical reactions within a cell. Okay, we have tons of reactions, or all the reactions we have learned before in the flax can be realized in the cell under what? Under very nice condition, room temperature or body temperature, or close to neutral pH, and very mild. You don't need heating, you don't need a special acid or a base to catalyze. This can be done within a cell, and all because of enzymes. Okay, all because of enzymes. So this chapter we're going to discuss what are enzymes and how do they function, and what are the factors affecting enzyme activity. Okay, first is what are enzymes? Enzymes, again, they pronounce enzymes, are proteins. They are biological catalysts, or sometimes people call them biocatalysts. Okay, what are catalysts? Catalysts basically accelerate or speed up chemical reactions. Okay, we do have catalyst in a flax as well. Okay, if you guys remember in chapter 2, we talked about hydration of alkene, adding water to alkene. We need acid as a catalyst, and in that case, acid is a catalyst. Of course, there's talking about inside a cell, in the flask, but here we're talking about inside the cell. Okay, catalysts, enzymes are biocatalysts that speed up a chemical reaction. Okay, this is a picture showing one of the enzymes which hydrolyze a disaccharide. This disaccharide happen to be sucrose. Okay, the table sugar we eat actually is being hydrolyzed by this enzyme. Okay, enzyme. Now, this word enzyme is derived from a Greek word meaning in yeast, meaning what? These are inside cells, and people have been using catalysts for a long time ago. Okay, especially utilizing yeast. Okay, or when people first realized they can make wine or liquor out of what of grapes, sugars, and the fermentation reaction is basically taking advantage of enzymes in yeast. Okay, all biological process require an enzyme or a few enzymes to complete, to sustain life. Okay, again, because all these chemical reactions have to be done in a very fast and efficient way, and also in very mild and ambient condition. Now, how does enzyme or catalyst work? Okay, what is the basic principle of these molecules that speed up chemical reactions is showing on this chart. Okay, in order for a reaction to happen, reactants, in order to become products, they have to first have sufficient energy to overcome a barrier. We call that activation energy or energy barrier. So in order for reactants to become products, these molecules have to collide, have to have sufficient energy to overcome the barrier. Okay, that's the energy required. And that energy mostly, again, coming from what? Molecules colliding with each other. Molecules are moving around each other a lot. But with the help of an enzyme or catalyst in general, the barrier will be lowered. The reason is most enzymes actually participate in the reaction, providing an alternative path. So the barrier of the new reaction with the help of this enzyme is much lower than before. So think about it, if the barrier is lower, the reactants will be overcoming the barrier what? Easier, because the barrier is lower. And because overcoming easier means what? More reactants can become products. And that's basically how enzymes work, or all in general, all catalysts work in this way. They speed up the reaction by what? By lowering the activation energy. We call the energy barrier for any reactions. Okay, that's just basically how enzymes work. Of course, the new pathway is involving enzyme. Enzyme mostly will participate in a reaction. Like we said, enzymes are proteins. Okay, proteins. And based on that fact, we classify enzyme into two types, just like we're classifying proteins. Enzymes, okay, are proteins. If the enzyme only has a piece of protein, nothing else, we call those enzymes simple enzymes. Okay, simple enzymes. Just like proteins, we call them simple proteins. But many enzymes, I would say most of the enzymes, contain not only the protein part, but something else. We call those enzymes conjugated enzymes. Most enzymes are conjugated enzymes. And for conjugated enzymes, the protein part is called the apo enzyme. Okay, apo enzyme. The other parts, we call them cofactors. Okay, cofactors. Cofactor and apo enzyme, they combine, bound together to give us a what? A complete conjugate enzyme. Let me ask you guys first. Cofactors is basically a what when we study the chapter of protein. Remember, protein are classified into simple protein and a conjugated protein, right? Conjugate protein has the protein part and something else, right? That something else is basically the cofactor in enzymes. So cofactor is basically the what of a protein? Prostatic groups, right? If you guys review the chapter 9, cofactor is basically the prostatic group of a protein. But now we're talking about enzyme, not protein, because the function of the protein is enzyme. So we call that apoenzyme, which is the protein part, and we call the prostatic group what? Cofactor. Very good. Okay, cofactor. Cofactors can be divided into two types. Inorganic ions, like metal ions, magnesium, zinc. It's very common in our system. Or complex organic molecule. Okay, complex organic molecule. So cofactor can be inorganic or organic. When the cofactor is a complex organic molecule, we usually call it a coenzyme. Okay, it is a cofactor, but we usually call it a coenzyme when it is an organic molecule. What are coenzymes? Most vitamins we take, especially vitamin B group. I don't know if you guys learned nutrition or AMPs talking about. Vitamin B, we have what? B1, B2, B6, B12. Those vitamin Bs are mostly used as what? Coenzymes. Okay, coenzymes, which are organic cofactors. And some metal ions, these are the ones that we see using as cofactors, inorganic cofactors like magnesium, zinc, manganese, and iron. Okay, sometimes chlorine can also serve as a cofactor. Does that make sense? This is the basic construction of an enzyme. Okay, conjugated enzyme has apo enzyme and cofactor. When cofactor is organic, such as vitamins, we call this cofactor what? Coenzymes. Make sense? Okay, next. Enzymes. Okay, enzymes have a lot of names. Okay, different enzymes are named differently, but all enzymes, or at least when you see a name, you can tell it is an enzyme by your first look. Because all enzymes, their name ends with 3 letters. A-S-E, pronounces ACE. Okay, pronounces ACE. All enzymes have a suffix of A-S-E. That means those are enzymes. Enzymes, because there are so many of them. Okay, remember, every single reaction in our cells is catalyzed by one enzyme. Because there's so millions of reactions, millions of enzymes, we classify enzymes by the type of the reaction they catalyze. Okay, by the type of reaction they catalyze. So we add a prefix in front of the ACE. And that prefix tells us what reaction they're catalyzed, what type of reaction they're catalyzed. For example, oxidase, it means what? Catalyze oxidation reaction. Hydrolase, it catalyze what? Hydrolysis reaction. Very good. That's what we have learned. Okay, of course we'll study the main types, but they're basically named this way by what? By the type of reaction. And sometimes, okay, in this class we don't talk about that in detail. We also add the substrate. That means they catalyze what? They catalyze, for example, if you catalyze oxidation, oxidation of what? We put the name of the substrate. We call the reactant, the enzyme acting on. Put the substrate name in front of the enzyme name to give you the complete name of the enzyme. For example, here, glucose oxidase means what? The enzyme oxidizes glucose. That means glucose is what? Is the reactant, and we call that reactant the substrate. Okay, we'll mention that name a lot in the future. Does it make sense? Okay, now, like I said, we don't talk about the substrate a lot in this class. Okay, we don't deal with the individual enzymes with their particular substrates, but we're going to study systematically the 6 types of enzymes, or the 6 reaction types of enzymes in this class. Okay, those are the basic classification of enzymes into 6 categories. So let's first take a look at these 6 categories. Okay, category. The names are listed on the first column: oxidoreductase, transferase, hydrolase, lyase, isomerase, and ligase. Okay, ligase. These are the 6 types of enzymes. Again, these 6 types of enzymes indicate what? 6 types of reactions they're catalyzing. Okay, you can see they're all ended with what? With ACE. Okay, with ACE. On the third column, the one I put in red, it's some clues for you to identify. For example, if you see a reaction, if you think in general, hey, this is an oxidoreductase, for example, then you look for it. Does the enzyme, does one of the description over here, for example, does the enzyme introduce a double bond? Does the enzyme remove 2 hydrogens? If it does, then you say, hey, this is what? Oxidoreductase. So basically, the third column tells you what the enzyme does, why it is classified as a certain type. Okay, for example, here, lyase, it does what? Remove of water, remove of CO2, remove of NH3, addition of water. These are the things lyase does. So these are some clues for you to, hey, I can tell what type of enzyme it is based on what the enzyme does, based on what the reaction shows. Okay, we can go back later, but I just want to explain how you use the table, but we will explain these in very details. But first, you have to know all these 6 types. Not only you need to memorize those 6 types, you have to be able to write them down, because you are going to write them down, the 6 types. Okay, oxidoreductase, transferase. Okay, that word, transferase means what? Transfer, something transfers. The enzyme does some transfer. Okay, hydrolase, again, hydrolysis. Lyase, we're going to explain what lyase are. Isomerase, and ligase. Okay, ligase. 6 types. So let's take a look at 6 of them, and I will give you reactions for every category. The first type, oxidoreductase. Okay, the name, based on the name, you know this enzyme does what? Oxidation and reduction reaction. In chemistry, no matter in biochemistry or just general chemistry, as long as it's in the scope of chemistry, reduction and oxidation happens at the same time. There's no way you can have one process without another. They always come, and that's why the enzymes called oxido and what? Reductase. Okay. Normally for oxidoreductase or oxidation reduction reaction, what we will see is what? Removing hydrogen, that's oxidation. Adding hydrogen is what? Reduction. We studied that in previous chapters. For example, in this case, from left to right, guys take a look. This is an alcohol, if you can remember, right? You see that? Again, you don't even need to know what the substrates are. You just need to know, hey, what's going on? What happened from left to right or from right to left? Right? On the left, it is what? Alcohol. On the right, we get a what? A ketone. Is that right? You see that CO double bond with 2 carbons. It is what? A ketone. Alcohol becomes ketone. What happened is what? These 2 hydrogens are what? Removed. You put a double bond. Okay, we learned this reaction. It's a secondary alcohol actually. Gives you a ketone. We learned that in reaction in chapter 3. This reaction is called what? Oxidation reaction. Removing hydrogen. And of course, the reaction can be go backwards. Or you add hydrogen, convert a ketone back to what? Alcohol. Okay, the forward is called oxidation, reverse is called what? Reduction. So this enzyme, no matter what the enzyme is an oxidoreductase. That's all you need to determine. Okay, you don't need to name the enzyme as long as you can determine the type of enzyme. And another important clue about oxidoreductase is, I remember you mentioned the very beginning of this semester, you guys were talking about NAD plus and NADH, right? Normally for oxidoreductase, if you remove a hydrogen, you need somebody to what? To take the hydrogen. Who is the taker? NAD plus or FAD, right? You guys learned A and B. We're going to study in chapter 12 as well. NAD plus and FAD are both nucleotide derivatives. They're hydrogen what? Receptors. So NAD plus will become what? NADH. FAD will become FADH2. Okay, you guys learned that in. These are very typical for oxidoreductase reaction because if you're removing hydrogen, you need a hydrogen acceptor. If you're given hydrogen, you need what? Hydrogen donor. These are accompanied with oxidoreductase reaction. Right? So that's the first one. Removing hydrogens or taking hydrogen. Okay, NADH and FAD are both coenzymes, basically. Okay, they must be there to take the hydrogen. Next, transferase. Okay, what is transferase does is what? Transfer something onto a molecule. Okay, in this case, very commonly seen is a transferase that transfers a phosphate from a molecule called ATP, okay, adenosine triphosphate, onto the sugar. You can see that this is alcohol, phosphate. It didn't put the whole structure of the phosphate, but you're making a what? Phosphate ester. So you're transferring a phosphate from ATP to sugar. And the ATP losing a phosphate becomes ADP. T stands for tri, D stands for di, means what? One phosphate is taken away, transferred to what? To a sugar. Okay, this is a very common transferase reaction. Again, don't worry about the name of these enzymes. Okay, all I want you to know is the 6 big types of enzyme. Transferase basically transfer what? A small part of molecule from one molecule to another. Okay. Phosphate transferase here. Transfer a phosphate from ATP is a very common reaction. Okay, that's it. Transferase. Third type, hydrolase. And you guys know hydrolase does what? Hydrolysis. Okay, hydrolysis. What's hydrolysis? We have learned many hydrolysis. Hydrolysis of ester, hydrolysis of amide, hydrolysis of alcohol, right? Carbohydrates. What does hydrolysis do is what? Cleave a bond, then add what? Water. Add water. Right? Cleave ester bond, add water, HNOH. Cleave amide bond, add water, HNOH. Here, cleave the glycidic bond, add what? Add water too. Okay, that's a hydrolyzed reaction. And you can put a side note here. Most digestive enzymes. What are digestive enzymes? Means what? Help us to what? Digest food. Okay, no matter digest protein, lipid, or carbohydrates. They are mostly what? Hydrolyzes. Remember, you eat protein. What is protein? Amide, right? A lot of millions of amide. If you want to digest protein, you have to cleave the amide bond by what? By hydrolyze. If you want to digest starch, what do you need to do? Chop off starch into what? Into glucose. You need what? Hydrolyze. If you digest fat and oil, you need to cleave what? Triglyceride. What are these triglyceride bonds? Ester bonds. You need what? Hydrolyze. So most digestive enzymes are hydrolyzes. Hydrolyzes. Next, lyase. Okay, lyase. I put here the most typical reaction for a lyase, that is adding or removing water from a substrate. Okay, this is different. This is different than hydrolyze. Hydrolyze is you have to cleave a bond, especially a bond in a large molecule, and adding water to make smaller ones. Lyase is basically adding water, most times adding water to a double bond, to change an alkene to a what? Alcohol. Or removing water from an alcohol to give you what? Alkene. Okay, it's not very typical, but again, I'll show you some tricks later on how to distinguish enzyme reaction. But this is the typical reaction lyase does, is to add water molecule onto a double bond or removing water to make a double bond. Next, isomerase. Okay, isomerase. Now, if you guys look at the name, ACE is enzyme, but the first prefix, isomer, means what? We learned this word, right? What is an isomer? Same molecular formula, but what? Different structure. So if you know what is isomer, you should be able to understand based on that the definition of isomer, and you should understand the function of this type of enzyme. This enzyme basically does what? Create isomers, converting one isomer to a what? To another. If you're converting isomer, what does it mean? It means both sides of the arrow or the reaction from A to B or B to A, doesn't matter. These two molecules should be what? Isomers. Does it make sense? Take a look. Do you see every component on the left? You can see the same component on the right. 3 carbons, right? 3 carbons. CO minus, CO minus. OH, OH. Phosphate, phosphate. They're what? Identical in molecular composition. But the difference is the structures are what? Different. In the molecule on the left, the phosphate is where? Here. You don't have to number it, but you know it's on this carbon. On the second molecule, the phosphate is where? Here. Not on this carbon, it's on this carbon now. So what happened? The switch places. Means what? You're making what? Isomers. Okay, isomers. So this is very easy to tell, in fact. Why? Because this enzyme, the reactant and the products are again what? Isomers. As long as you know what are isomers, you should be able to tell isomerase reaction. Sorry. The last type. Okay, the last type, ligase. Okay, ligase. This word you may not get familiar with. Ligase, the prefix LIG came from a word ligation. Have you guys heard of a ligation? Ligation. What does a ligation mean? Anybody knows? Okay, ligation means join, link something, Like you put two piece of, like your necklace is broken. If you put two open pieces together, join it. That step is basically, you can call it ligation. Means what? You join two parts together. You add one part to another. That's called ligation. So what ligase does is what? Forming a new bond between two substrates. Usually, this process needs energy. And ATP is the energy provider. Okay, you guys may have heard of it. We will study that in chapter 12 as well. And you can see that here, in this case, ATP only does is what? Provide energy. The phosphate is not transferred to anywhere. This is the phosphate. It's broken. ATP becomes ADP and a phosphate. The bond is broken, and the energy in that bond is what? Is used to do that ligation. This what happened is, you can see that this carbon is ligated with that carbon. Basically, the molecule add one extra carbon on it. Do you see that? Okay, this molecule, 3 carbon, add a carbon from CO2 to give you this guy. To now become how many carbons? 4 carbons. Like again, like I said, 2 parts joined together. And usually this reaction requires energy. ATP provides that energy. And that is the 6th type of enzymes. Good? Okay, something to summarize, and something I want you to know is, first, I doubt you will see any like a very uncommon, like a weird and hard to tell reactions, and or too far away from what we show from this class. So the one you will see in your quizzes, tests, and practice are very commonly one. Someone you can maybe decide and first look, as long as you understand these 6 types. And number 2 is, I cannot speak for the final, but in your 3rd test, I will give you 6 reactions. You only need to answer 4 of those. No more. I will emphasize that as well before the test. 4 means what? You choose the 4 you're comfortable with. The other 2, hey, I don't even know if that ligase, lyase, or hydrolyse. Leave it open. You only need to answer 4 to get full credit. Is that fair? I said you guys are not required to know a lot, but you need to know something about the classification of enzymes. That is basically how enzymes are classified. They're initially classified into 6 types. Good? Okay, again, we can practice more. You have more in your quiz as well next week. Okay, you have seen more examples. Okay, so very quick. This is the reaction showing the discoloration of some fruits, especially like apple, right? You cut it open, you will see that the apple surface will become brown after a few minutes, and that reaction is shown here. So the enzymes involved in this process are called a phenolase. Okay, you don't need to know, I don't need to know what phenolase is, but take a look at the reaction. What do you think the type of phenolase is? What type of enzyme called? Oxidoreductase, right? What happened is what? You can see that phenol is become what? Ketone, right? So hydrogen is what? Removed. Okay, of course, again, this reaction definitely has NADH or FAD serving as cofactors. Or in this case, you can see that eventually the hydrogen goes where? Oxygen. Okay, that's why you show oxygen here. Even though NADH and FAD, if they're involved, they're intermediate receptor. Eventually, where does the hydrogen go? Oxygen, right? The same for us. We breathe. What the hydrogen is used for? And what are the oxygen we breathe in are used for? Eventually, take what? The hydrogen to give water. Okay, you can think about where does the high oxygen? We breathe in, we use them, used for what? Accept the hydrogen in the last step. We'll see that in chapter 12 as well. Right? So, very good. That's the classification of enzymes. Okay, next topic is how does enzyme work? And what are the key terms or the key steps in a reaction of enzyme? And in order to explain the enzyme works, we first have to introduce a term called enzyme specificity. Okay, specificity. What does specificity mean? Specificity means what? Enzyme has to be what? Strict on something. Okay, they're not universal. Enzymes are strict on the reaction they catalyze. That's why we have so many enzymes for so many reactions. We don't just have one enzyme does everything. We have what? We have many cases one enzyme for one reaction. That's why we talk about specificity. Enzymes are specific. But with regard to specificity, we have different types or levels. One type of specificity is called absolute specificity. It means one enzyme can only catalyze one substrate in one reaction. Nothing else. It only does that one job. Just like one car, only take you to one place, nowhere else, nobody else. That's called absolute specificity. Another type of specificity is called relaxed specificity. It means the enzyme can accept substrates with certain similarities in their structures. Or you can understand it as enzyme, the car can take you or someone like you to that place. They won't just allow, they cannot just allow you to get in the car, or someone looks like you or dressing like you will still accept it. That's called relaxed specificity. That acts on structurally related or similar substrate in that certain reaction. Another specificity is called a stereochemical specificity, meaning the enzymes can only accept what? One enantiomers over another. And we actually have seen or have talked about that before. We know our body only can accept what? D carbohydrates and L amino acids. Why? Because our enzyme have stereochemical specificity. When we accept D carbohydrates, our enzyme will not accept L. They will not take the mirror image of the L, of the D carbohydrates. The same, our enzymes can use L amino acids to make our proteins, then the isomers are not taken. That's called a stereochemical specificity. Okay, this is very easy to understand because isomers are mirror images are totally different with enzyme environment. But in order to explain these two, why some enzymes are strict, very strict, why some enzymes have some tolerance, they are relaxed, we have to take a look, how does enzyme work? Okay, what is the step of enzyme catalyzed reactions? Enzymes, again, is a protein. Okay, protein is a large biomolecule, 3D biomolecule. But in an enzyme, there is a place at which the reaction takes place. Not the whole enzyme. There's a place, a piece of the enzyme, that where the reaction happens is called the active site of the enzyme. What the active site does is what? Is to takes in the substrate, the reactant, and then the reaction will happen where? At the active site. Okay, this is the scheme, shows you what happens in an enzyme catalyzed reaction. Okay, it looks very simple, but I want you to know very detailed. You can explain using word, and you put the image in your mind about the process. First step is the enzyme binds to the substrate at where? The active site. And the product here is called the enzyme substrate complex, or you can call it ES complex. ES stands for enzyme substrate complex. After it's bound, remember, enzymes are what? Proteins, right? At the active site, the amino acid here, some amino acid here, mostly it's side chains, here will help to do whatever it does. This is a hydrolase. It looks like a hydrolase. So it will help to what? Cleave the bond, add water. Okay, again, different enzyme active, but the amino acid residues, especially the side chains, actually does the reaction at the active site. So the ES complex will turn into a EP complex. P stands for what? Products. Okay, they're still bound, tight bound. That's why the amino acid here can function, does the reaction for you. And then after you get your products, the products will be released to give you a fresh enzyme, again, which can again what? Take another substrate. The enzyme is not done. Enzymes that can go back to work again. That's why what? We don't need a lot of enzymes in our system, right? Because enzyme can be what? Recycled. And they do this, of course, fast. We talk about this what for 5 seconds. For 5 seconds, they probably have done millions of reactions already. Okay, millions of reactions. And this is, again, the very basic, the very fundamental mechanism of reaction. Easy, right? But again, I want you guys to be able to explain using the scheme. You don't have to draw the scheme, but you have to know, okay, this is what happens. Active site, what is active site? The bound, EP, ES becomes EP, and then products released, enzyme can what? Can start again. And we said enzymes are proteins, right? How can a protein bind a substrate? It's because on the active site, again, we know on the active site we have a lot of what? Amino acid. And amino acid have what? The side chains are exposed outside, if you guys remember chapter 9. And those side chains will what? Will interact with the groups, whatever the groups on the substrate. So the interaction, the binding is very similar to tertiary interactions of proteins. Okay, here is the picture shows you. This is a mimic, not a real enzyme picture, but show you how the substrate is bonded to the active site. You can see that the group on the substrate are actually doing what? Interacting with the side chain of what? Of the active site. Okay, this is the active site. There's some side chains here, and the groups on the substrate are actually interacting with them, so the substrate can what? Can dock into the active site. And the interactions are here. Do you see the names from here? They're what? From tertiary interaction, except disulfide. Normally you don't see disulfide, but you see electrostatic interaction, you see hydrogen bonding, and you see hydrophobic interactions. Okay, there's a very common when enzymes bond substrate. Okay, again, this is another real picture. Here I want to show you a real picture of an enzyme. Okay, this is I think it's an isomerase. I forgot. It's an isomerase, yes. Picture, and this is the sugar. This is a sugar molecule here, and this is the active site. You can see how beautiful the picture is. Okay, you can see the hydroxy group is interacting with the side chains. And here is a metal ion, manganese. What is manganese called? If the enzyme has a metal ion manganese, this is called what? The cofactor. You see that? This is what cofactor is doing here, helping the binding of the substrate to the active site amino acid side chains. Okay, when you see this, you find nature is amazing. How this all happen. It's just one enzyme showing you how molecules are docked into the active site, and of course the reaction will happen afterwards. Okay, again, this is the substrate shown here. That's the cofactor. These are the active site amino acid residues, the side chains. Again. Okay, now after you understand the basic action of enzyme. Now we want to explain why enzymes have either absolute specificity or relaxed specificity. Some enzymes, the active site is very rigid. It's very rigid. And because of the rigidity of this active site, some enzyme can only accommodate a specific substrate that can perfectly what? Bind with it. And we call this type of binding key and lock model. Okay, or key and lock and key theory. What is lock and key? The key must match the lock perfectly. No mistake. They bind perfectly because the active site is what? It's rigid. The enzyme is relatively rigid. Okay. Another theory, which is actually more common in biological system, is the enzyme active site is not actually fixed in a certain shape. When a substrate approaches, the active site can actually change a little bit to accommodate the substrate for binding. And this theory is called induced theory. What does induced mean? When you come in, I can accommodate you to what? To take you in, to bind substrate. And because of the flexibility of the active site, the enzyme can take what? Substrate of similar structures, because they can change a little bit. Of course, you cannot have big structural difference. The substrate that takes in still have to be what? Have to be similar. And we call this theory induced theory. The enzyme active site can adapt a little bit to take substrates with similar structures. Does it make sense, guys? Good? All right? Move on. So far good? Any questions?
For the substrate we talked about, if the substrate is a protein, what if the substrate is not a protein, it's say a glucose or carbohydrate, then how does the interaction happen?
What do you mean happen?
Like how does the interaction happen? Because we
the substrate can be small molecules or protein, doesn't matter. What changes here is the active site is changing. The substrate doesn't change.
Yeah, but we talked that proteins have R groups.
Right.
And that is what interacts.
Your small molecule also have groups on it, right?
Okay.
Take a look. This one is the one I showed you. This is a small sugar.
Yeah.
This is a 5-carbon sugar actually. Sugar has what? Hydroxy groups.
Yeah.
So what happens here is this hydroxyl group of the sugar bound with the active site of the side chain of the active site through what? Through the cofactor. So this is showing how small molecules are used. Very similar. Okay, if you're protein, then the protein of the side chain of the protein interacting with the side chain of the active site. If it is a small molecule, then the groups on the small molecule interact with the amino acid on the active site. Either way, yes. The interaction is similar to tertiary interaction, but the substrate is not limited to proteins. Could be a small peptide, could be a protein, could be a nucleic acid, but they all have functional groups, and the functional groups will what? Interact with the side chains of the active site. Does it make sense?
Yes.
Okay, good. Any questions, guys? Anything about enzyme functions? All right, next is something we will use in our following lab. Okay, following lab. We're going to discuss factors that affect enzyme activity. Okay, or enzymes are regulated by these factors. Okay, are affected, influenced by these factors. And we're going to investigate these factors in our lab. Okay, there are a few factors. That's why I mentioned in this class, you guys will be responsible for one factor per group. And you cannot mess up, because if you messed up, we don't have that part of the data. We don't know what's going on. And I guarantee you, if you don't mess up, you will find out the chart you put your graph will be very close to the chart we have seen in the class. You're going to make these charts. Okay. This is a very nice lab. You can and actually prove, oh, you're not showing us from textbook, you're actually making this chart. You will see that. All right, so let's take a look. What are the factors? The first factor, temperature. Okay, temperature. Each enzyme has a temperature that the enzyme acts the best. Acts the best means what? The enzyme will have the highest activity. And that enzyme, that temperature of the enzyme, that temperature is called the optimal temperature of the enzyme. Optimal means what? The best temperature. The temperature that the enzyme is the highest activity. And you can see that in the graph here. At the optimal temperature, the enzyme have what? It's the peak, highest activity. For our enzymes, for example, human enzymes, the temperature is around 37 degrees Celsius. That's our body temperature. Okay, that's your body temperature. Anywhere temperature lower than the optimal or higher than the optimal temperature, the activity is going to what? Decrease. Okay, decrease. But take a look at this chart. It's very unique, and also we're going to compare with the chart next page. At high temperature or extremely high temperature, the enzyme activity is going to go back to what? Or go to what? Zero. Why? Because we know heating is a physical factor that will cause a protein to what? To denature. Right? Like you're cooking an egg, the protein is basically denatured. And if a protein is denatured, the enzyme is what? It's basically gone. The function is lost. So at high temperature, your enzyme is potentially will be denatured. But at low temperature, your enzyme activity is going to decrease, but will not denature. Right? We never said cold temperature can denature an enzyme. It wouldn't. Okay, it won't. In fact, in our labs, in a lot of labs, bio labs, they store enzymes in minus 20 degrees in the fridge. And a lot of living things, can survive what? An extremely cold temperature. Not many things can survive at an extremely hot temperature. We did find some very thermal bacteria near the volcano, the sea volcano nearby. But those are extremely rare bacteria there. Okay, but most enzymes won't tolerate high temperature. Low temperature is okay, even though the activity is low. Okay, so that's a very difference in this chart compared to our next one, the pH. Okay, what is pH? pH is the what? The scale evaluating the acidity or what? Basicity of an environment of an enzyme. Of course, very similar to temperature, each enzyme has a what? Optimal pH at which the enzyme has the highest activity. Now, we know our temperature for our enzymes, the optimal temperature is all around what? 37. No matter where you are, your temperature, your body temperature is very well maintained. But optimal pH of our enzymes is not a universal. It's not the same. It's dependent on what? Locations. Okay, where the enzyme is located. For example, our stomach enzyme. Okay, most of the hydrolysis in our stomach. The pH, optimal pH is around 2. Very acidic. Why? Because our stomach fluid pH is what? Is 2. Okay, or the enzymes in our small intestine, their optimal pH is around 8 because our small intestine is slightly basic. Okay, here, this chart shows you the optimal pH of different enzymes found in different species or different locations. Okay, just show you, hey, not all enzymes have the same optimal pH. Depends on where the enzyme is located, in a species or in the same system. Now let's go back. Also similar to optimal temperature, the pH is the activity of the enzyme is going to decrease as you what? As you deviate from the optimal pH. Okay, the chart you can see is similar, but the difference is at extreme pHs. When you have a extremely low pH or extremely high pH, what do you see here? The enzymes will both become what? Denatured. Because strong acid and strong bases are both factors that will cause a protein to denature. So the chart for pH is more of a, I shouldn't say symmetrical, but more regular bell-shaped chart. But the chart for temperature is not. And we know why. Because low temperature does not necessarily cause enzyme to what? To denature. You guys see the chart difference? Again, you will make your own charts. We will do temperature, we will do pH in our own labs. And we're gonna make a chart very nice. All right, so that's the second factor. The third factor is substrate concentration. Substrate concentration. What is substrate, guys? What is the substrate of an enzyme?
The site where the enzyme is active.
The what? The reactant, right? The molecules the enzyme is catalyzing into what? Into products. So even without looking at the chart, you can imagine when you have more substrate, the enzyme will have what? More reaction to what? To catalyze. So the activity of the enzyme will what? Will increase. Of course, you have more guys to catalyze. You have more reaction, turn into products. But if you take a look, the chart is not all straight on. The chart is actually what? Curved. Means what? When you increase the substrate concentration, the increase of the reaction rate is going to what? Slow down. Eventually, flat out. Is that right? Why is that? Remember, enzyme has what? Has active site. When you have a lot of substrate, increase a lot of substrate. If all the enzymes are occupied, what happens? If you keep increasing the substrate, there will be no more enzyme available to what? To take your substrate. At that point, the enzyme reached the maximum velocity. That is why you see the plateau, the flat out. Because at that point, your enzymes are all what? Occupied. All seats taken. You have to wait somebody to get up. It becomes what? Before new guy can come in. So, the substrate affecting enzyme concentration curve, the chart is curved. It means the increase of substrate concentration, the enzyme activity is going to increase fast, but gradually will what? Slow down. Eventually reach what? The highest. This is the highest activity. Okay, this is the highest activity. If you keep increasing, then we'll just remain flat. Why? Because you have no more enzymes available. This is substrate concentration is a factor. And we're gonna make this chart too. One group will make this chart too. Next, okay, here, I'm sorry, this one is the chart showing you different enzyme has different number of seats or how fast they can turn over. Okay, some enzymes are slower, some enzymes are faster. Depends, but in general, you can see how amazing, how amazing enzyme can work on our reactions. Okay, this is a concept. Okay, we won't talk too much, but this is a concept called turnover number. Turnover number means how many molecules they can catalyze per minute. Okay, some enzyme like this guy, carbonic anhydrase, can catalyze over 36 million molecules per minute. Means what? As long as your molecule is within 36 million per minute, they can keep increasing. But some enzyme like our DNA polymerase, they're making large molecules, they can only catalyze 900 molecules per minute. Depends on the type, but you can see how efficient our enzymes are, how many molecules they can catalyze per minute. Okay, but again, enzyme will reach maximum velocity when you have too much substrate. Okay, the last factor, is enzyme concentration. Okay, enzyme concentration. And this chart is very unique because it's what? Straight line. Straight line means what? When you increase enzyme concentration, the activity is going to increase as much as you want. It's very easy to understand because you have more what? You have more enzyme to what? To catalyze the reaction. You have unlimited amount of seats to increase. Then anybody can come in to catalyze it, can come in to have the reaction done. So enzyme activity increases as the enzyme concentration increases. Okay, it is a straight chart. So these are the four factors. Okay, four factors we're going to investigate. Okay, actually, in fact, we only investigate 3 of them in our labs. Okay, and another factors is something we'll talk about next Monday. All right, do you guys have any questions? I think we have time to finish this chapter, but I'll save it to next Monday. Then we'll what do you think? You want to finish this chapter today, and then next Monday we start a new one, or finish the second half and start half of chapter 11 Monday? Let's finish today. It's be a more complete story. You'll have a better idea about enzymes. Okay. The last part of this chapter is enzyme inhibition. Okay, enzyme inhibition. What does inhibit mean?
Slow down.
Stop, slow down. So enzyme inhibition means some substance that can bind to the enzyme to what? To slow down or even completely stop the enzyme activity. That's called inhibitor of enzymes. Okay, inhibitor of enzymes. Okay, this part is very interesting. We have a lot of medicines developed based on enzyme inhibitors. You can think about it, inhibitor does what? Stop or change the enzyme we have. A lot of reactions we want to slow down, or a lot of reactions we want to stop happening for disease. Enzyme inhibitor will be a good way to do that. Now, depends on the actions of the inhibitor. We classify the inhibitor into 2 types: irreversible inhibitor and reversible inhibitor. By the name, you can easily understand what is irreversible means. Means you cannot turn back, right? Reversible, you can what? Reverse. Okay. More importantly is why? How? Why some you reverse? Why some you cannot reverse? For those reversible ones, how can you reverse? So these are something we want to talk about now. So first, take a look. Irreversible. Again, irreversible means you cannot what? Turn back. Means the enzyme is technically what? Dead. Lost its function completely. Why is that? The reason irreversible inhibitor can completely stop the function is the inhibitor makes a covalent bond with the amino acid side chain on the active site. Okay, remember we said the reaction happen has to be something to do with what? The side chain of the amino acid at the active site. Irreversible inhibitor makes a bond, covalent bond, like the one we see in this class. Then what? Then the enzyme lost its function. And covalent bond is very strong. Normally you cannot cleave the covalent back to the original enzyme. That's why the enzyme is what? Is lost, is irreversible function lost. Okay, this is a picture showing the irreversible inhibitor of a bacteria enzyme, not ours. Okay, this enzyme is called transpeptidase. Okay, transpeptidase. Basically, this enzyme is responsible for making the bacteria cell wall. But penicillin, the antibiotic, make a covalent bond with the amino acid serine on the active site of this bacteria enzyme. Okay, you actually have seen this bond. You can actually understand this reaction. This is the amide bond. You guys see that? CO1. And this is called beta-lactam antibiotic. And the amide bond is opened. They make a water bond. You can see that? This is the alcohol, OH. You see, this is the bacteria enzyme active site. They make a what bond? COO. What is it? What is the bond? COO. This is a new bond. What is COO bond called?
Acidic?
No, the bond is called what? CON is called amide, right? COO is called what? Ester bond. Okay, the CO make a bond with the oxygen with an ester. And because the active site amino acid is making a bond with the inhibitor, the enzyme is permanently what? Inhibited. Does it make sense? Okay, this is irreversible inhibitor. Okay, why? Again, covalent bond to the amino acid side chain at the active site.
Is this why the doctors they ask you to take probiotics when they give you an antibiotic?
Take you what?
To take probiotics. Sometimes, with children, they would say just give them probiotics.
No, I don't think most antibiotics sometimes will cause diarrhea, so they probably ask you to take a probiotic. I'm not exactly sure.
Because they say that some antibiotics they kill the bacteria inside your gut.
Yes,
So-
the same. We have a lot of good bacteria in our internal a digestive system, and penicillin is not a specific one. It's universal. Sometimes kills your own bacteria, so that means your intestinal digestive system loses balance. That's why it causes some digestive problem or diarrhea. That's why you take some prebiotics to supplement.
Because that kills the bad bacteria and the good bacteria that you need and cause you that.
Correct.
Gives you whitus where you bleed out your stomach.
That one I'm not a medical.
Not with penicillin.
Now, another example of irreversible inhibitor is shown here. Involves a nerve gas we have talked about in chapter 5 and chapter 6. Okay, the nerve gas, such as this one, is a bioweapon that can covalently bound with our enzyme, okay, mostly acetylcholine hydrolase. So you cannot hydrolyze your choline, you will keep contraction, for example. Okay, the same mechanism. That's why these biogases are very dangerous or high impact power, because they irreversibly shut down your enzyme. There is no antidote. Okay, there's no antidote. Okay, this is another example. Now, another type of inhibitor, reversible, again means what? Means they're not making covalent bond with your enzyme. So your enzyme has a chance to go back. When the inhibitor what? Dissociate from them, just go away. Okay, go away. Now, depends on where the inhibitor binds, which part of the enzyme the inhibitor binds. We further classify reversible inhibitors into two types. Okay, into two types. Competitive inhibitor or non-competitive inhibitor. Okay, let's take a quick look. I'm glad I finished this part. You're gonna use all these to finish your lab, the post-lab as well, in fact. Competitive inhibitor binds at the active site, not covalent bond. Binds at the active site. And now you know why it's called competitive. When the competitive binds in the active site, what happened? Your substrate what? Has no place to bind, right? Because someone take the seed. Your substrate cannot get in. That's why they're what? Competing. That's why this inhibitor what? Is called competitive. Now, quick question. When a competitive inhibitor binds to the active site, what can you tell between these two, the structure-wise? We know substrate binds the active site. And now competitive inhibitor also can bind to the substrate. So what can you tell? The structure between the structure of these two. They're similar, right? Competitive inhibitor normally resembles what? The substrate. Or they're designed to resemble the substrate. Okay, that's how can we tell the structure of a competitive inhibitor. They normally resemble the structure of the substrate. And they compete with the substrate. So your substrate will have what? Will have no chance to bind to the active site. And because it is reversible, means the substrate can be what? The competitor can be what? Kicked away. How? Increase the concentration of the substrate. Then they will what? Kick it out. Again, the action can be reversed by increasing substrate concentration. Right? You get more body to compete with you. So it depends on who is competing, who wins more in amount, in concentration. So a few terms again. Competitive inhibitor binds to where? The active site. The reason it can bind to the active site is because what? It resembles the substrate. And this action can be reversed by what? Increasing the concentration of the substrate. Non-competitive inhibitor, this is what we have just shown, not yet, non-competitive. Okay. Now, because the substrate structure and the active site structure provides some useful information that we had actually designed inhibitor if we want. Design what? How do we design inhibitor? We design something what? Similar to the substrate. Okay, this is exactly how or a lot of drugs are made. Okay, for example, here, sulfa antibiotics. Okay, sulfa drugs, they're a very old generation of antibiotics. Okay, this is I think maybe the first sulfa drugs made. Okay, this is the substrate. Again, this is substrate for a bacteria. We're designing inhibitor for bacteria. Okay, this is the p-amino benzoic acid. This is the sulfa drug. Do you see they're similar in structure? See that? Not the same, but what? Look similar. So this guy serves as a what? As a non-competitive inhibitor for this enzyme. By the way, this is the acid used for bacteria to synthesize folic acid. If they're inhibited, they cannot synthesize folic acid, then the bacteria will be inhibited. The growth of the bacteria will be inhibited. So this is how we designed sulfa drug. Okay, they're still using sulfur drugs nowadays. They're different one, but they're again structurally they're related to this one. Why we related to this one? Because this one resembles what? The natural substrate of the bacteria. Okay, so that's a non-competitive, that's a competitive inhibitor. Another type of reversible inhibitor is called a non-competitive. Non-competitive inhibitors does not bind to the active site. It binds to somewhere else. Okay, binds to somewhere else. When it binds to somewhere else, the active site will change its conformation, like shut down by itself. So your substrate what? Cannot get in. Okay, cannot get in. Okay, someone wonder why there's this action like this. Usually, if you have a non-competitive inhibitor, this guy serves as a regulator for an enzyme. What does regulator mean? Sometimes, hey, I want to slow down the reaction. What do? I add something to the enzyme so the enzyme what? Close its seat, close its door temporarily. Then I can what? I can regulate the speed of the reaction. So a lot of times, non-competitive inhibitor serves as a regulator. And that type of regulator is called allosteric regulator. On the left picture, this is the allosteric regulator, also a non-competitive inhibitor. When it binds to the enzyme, what happened? You can see the active site what? Changed. So the substrate cannot what? Cannot bind in. So the reaction slowed down. Right? And in comparison, in our system, we do have some positive inhibitor, like a regulator. When this thing binds in, the enzyme actually opens up. Okay, that's called a positive allosteric inhibitor. But again, they're all regulators. How the enzyme got regulated. Now let's come back to the negative regulator, the non-competitive inhibitor. I'm going to explain to one very common and classic inhibition in our biological system that is very useful and also perfectly demonstrate this regulation. And that regulation is called feedback inhibit. Okay, feedback inhibit. Okay, I'll finish here and I'll leave the last part for you to read. It's very easy to understand as well. Okay. Usually in our biological system, the reactions are carried stepwise. So we don't have one single reaction. Especially in our body, like a metabolism process, all these reactions are like what? Stepwise, from A to B, B to C, C to D, and we have maybe 10 or 20 steps for a product. So imagine if you have a sequence like this, from A to B, B to C, C to D. D is our product, the one we want. And of course, each step we have what? One enzyme. How can we tell, hey, I have a lot of product, you guys need to stop. Don't make more D anymore because we have good amount. We need to slow down or stop. Our body have a very classic way of doing that. It's called feedback regulation or feedback control. In this regulation, the final product will serve as a what? Inhibitor for the first enzyme. Think about it. If this is the final product, this is the first enzyme, what it does. If you have too much of the product, then the product will what? Bind to the first enzyme, and then the first enzyme what? Shut down. If the first enzyme shut down, the following step what? Shut down too. So you don't waste of synthesizing B and C, right? You stop in the first step. Now, after this shutdown, we have a lot of D, but D is getting used in our biological system. So when the D concentration drops down, then what? Then the D will start to what? Leave here. When D leaves here, the enzyme will go back to what? Work again. Isn't that beautiful? Okay, that's called feedback control. The product of the final step serves as the inhibitor of what? Of the first enzyme. If you have a lot of it, the enzyme will what? Slow down. If you need more, then that means you don't have a lot of it, the enzyme will what? Go back. That's why it's called irreversible. Reverse means what? Depends on how much you have this guy. If you have a lot of this guy, inhibit, slow down. If you don't have a lot of this guy will dissociate, the enzyme will what? Go back to work. Okay, again, nature has a way of evolution, has a lot of these things that you would think is very amazing. Okay, once you stop the first enzyme, again, you're not wasting B and D, C staying there. You just, because you stop what? The first step. That's called feedback control. And D is called what? The irreversible inhibitor of the first enzyme. Or we call negative allosteric regulator of enzyme. Does it make sense? Okay, again, I won't go over these, but I want you to kind of read very briefly what are zymogens, or we have time we can talk about later in our next week's recitation. Okay, zymogens are another way to regulate enzymes, and also a class of hypertension drugs are actually developed by zymogens. I want to give you an example too, but we can stop here.